I'm Leslie Kean. I'm a pediatric bone marrow transplanter. I work clinically at Seattle Children's Hospital in Seattle, Washington, and am primarily appointed at the Seattle Children's Research Institute.
My name is Ben Watkins. My appointment is at Emory University in Atlanta, Georgia. I have recently extended to Seattle, Washington to continue my research in the Seattle Research Institute with Dr. Kean.
Doctors Leslie Kean and Benjamin Watkins are among the authors of a recent plenary paper on transplantation.
To watch somebody's development from a trainee into an independent physician scientist is huge, and I think that's one of the joys of having a lab.
I was very fortunate to be able to join Dr. Kean's lab 5 years ago and have had incredible mentorship. It was wonderful to get a plenary paper, especially at this phase where I'm transitioning into my own career.
The first 4 authors in the paper are all trainees in my lab, and I get more out of my trainees than I'll ever give to them.
I think that's something that's very important, is finding a mentor that's not only knowledgeable and a good teacher, but also looks after your best interests.
My lab is very interested in graft versus host disease after bone marrow transplantation, and the idea of the study was to understand a new targeted agent for graft versus host disease. But as bone marrow transplanters, one of the key questions that we really struggle with in our patients is how they reconstitute their immunity after transplant. And one of the things that I think is really important when you run a clinical trial is not only to get that yes or no answer of whether your new drug will work, but also really to delve deeply into the biology of what you're studying.
A big part of the data management is one, having intimate knowledge of the patients that were on the trial. And so knowing every detail about each individual patient and keeping meticulous records on what at the time may not seem relevant, but you don't know what's going to be significant until you're actually looking at the data at the end.
My favorite thing is when I can sit on the weekend in front of my computer and just look at data. And this paper was actually conceived doing that. I was just really having a good time, as nerdy scientists do on the weekend, looking at all the data. And what jumped out at me was the central observation of that paper. So it was really one of those wonderful moments where what we saw was that our patients had really different pace and extent of immune reconstitution when we looked at a certain cell type, the CD8 effector memory cell. And so that was the first observation, and it really took 2 years in collaboration with a number of different investigators and laboratories and biotech to come to the answer we came to.
A big portion of my role was gathering all of that clinical data and working with Yvonne Susmuth in linking it to that laboratory data.
To make the discovery mean something, to really get meaning out of our first discovery, we needed to use technology that at the time was very new, deep sequencing technology for the T-cell receptor. So we partnered with a biotech firm, and they are authors on the paper as well. One of the surprising observations that we made is that at the same time that the CMV-specific T cells were expanded, you get this contraction of our naive T cells and also of the rest of the underlying repertoire. And we think that that may be actually critical in terms of causing patients after transplant that reactivate their CMV to have more profound immune dysfunction than other patients that don't reactivate CMV. So it provided an explanation for a mystery that really wasn't able to be addressed with current technologies, and we applied these new technologies to answer that question. The new horizon is for clinician scientists like Ben is to be able to think about the patients that we put on these trials and apply these really complex molecular techniques to them to really understand at a level that we previously couldn't how they're doing after transplant.
Maybe we'll be able to pick up signs of the disease before the disease occurs, and that may open the door to new novel therapies that could potentially prevent graft versus host disease from happening.
We are poised now to use techniques to answer questions that we previously were not even able to dream of.
Read more cutting-edge research in the current issue of Blood.